My name is Susie Theroux, and like Ali said, I'm going to be talking about our efforts to work on integrating molecular methods into bioassessment programs. And so my title posed the question, are we there yet? And so to cut to the chase, the answer is we're getting there. And so for this talk, I'll go over some of the different applications where we are closer, and some of the applications where we're still continuing to develop these molecular methods. So first off, what really is the key motivating factor for developing these molecular-based approaches for bioassessment? And that's really the fault of having really successful bioassessment programs. And so due to the success of these bioassessment programs, there's been really high demand for biological data. And this increased demand on biological data has resulted in something that we call a capacity limitation. So it just puts a really big strain on the taxonomists. And so that can result in time lags in getting biological data turned around, and it can increase costs for getting some of this important data for making management decisions based on this bioassessment efforts. So in order to provide an alternative to the traditional morphology-based or microscopy-based methods of generating taxonomy data, we've begun to develop some of these new molecular or DNA-based approaches. And some of the key selling features of these DNA-based approaches are that we have the ability to generate species data, hopefully quicker, more reliably, more repeatably, and hopefully also for less cost. So there are a number of different programs that are already pioneering the use of these DNA-based methods for bioassessment, and so I've listed a couple of them here. They range from everything from collecting organism data on algae, invertebrates, and fish. We also look for human health pathogens, potentially toxic algae species, and then also molecular methods are routinely used for surveillance and detection of endangered and invasive species. So there's really a wide range of different applications for these molecular methods. If you do not spend your days working with DNA, I thought it would be useful to provide a little bit of an overview of some of our favorite methods that we like to use in bioassessment when it comes to applying these DNA-based technologies. So the first one is called quantitative PCR, which, as the name suggests, is a great way to quantify some of your target organisms present in your sample. So using quantitative PCR or qPCR, we might ask questions such as how many microcystis cells are in my sample, or how many pathogen markers are present in my sample. qPCR is a great way to begin to answer some of those questions. If we're interested in actually identifying some of the species that are present in our sample as opposed to just quantifying a target DNA, we move on to DNA barcode sequencing. So with DNA barcode sequencing, it's a great option when you really want to know what is the species of fish that I caught, or is this a quagga or a zebra mussel that I have. And if you're interested in identifying beyond just a single organism and you want to look at whole groups of organisms or a consortium all at the same time, then we move on to DNA metabarcode sequencing. And so this is a great option when you want to see what the whole biodiversity of fish are present in your sample, or if you want to look at all the different invertebrate species present in your sample. So for today, what I'll be doing is just stepping through each of these different approaches, some of the common ways that we apply them, and some of the main pros and cons or limitations of the technologies as they stand now for being incorporated into bioassessment. So first off, quantitative PCR. As I said, this is a really great way for quantifying target genes. One thing that you really do need to know, though, is who you're looking for. So it really does rely on having specific probes or primer sets for targeting your organism of interest present in your sample. Already, though, quantitative PCR is being routinely used for a number of different monitoring programs that might be looking for delta smelt or microcystin or pond turtle. So there are a number of different organisms from microbes to megafauna that we use qPCR for monitoring in aquatic environments. And one of the other cool things is that some of the newer technologies, such as droplet digital PCR, are super sensitive, and so they can actually detect the target or the gene of your organism that you're interested in surveying at super low levels, which is really great, again, if some of these organisms are especially rare or difficult to detect out in the wild. Some of the main limitations of quantitative PCR right now is that the quantity of your target genes present in your sample does not always equal the quantity of your target organism for all species. And a lot of that has to do with biomass variation in your organism of interest. So if we're looking for a single cellular organism, we can be looking for a gene that occurs just one time in its genome. But if we're looking at a multicellular organism, such as a fish, the amount of DNA that's sloughing off of that organism is represented in the quantity of that target gene copy in your sample. And so we can't always get an exact quantification of our target organism by using the quantitative PCR method if we're dealing with high biomass species. There are some ways to try and correct for that, different in concentration, but they're really kind of on the vanguard of using these methods. Additionally, one of the big limitations of the quantitative PCR approach is that there are a lot of questions about the fate of DNA in the environment that are still poorly understood. So that has everything to do with, once the DNA leaves the organism, where does it go? Does it travel downstream? Does it glom onto some organic matter present in the sample? Does it get degraded by UV radiation or temperatures or different microbes that are digesting it? And so all of those different processes impact the longevity of that DNA present in a water body and can impact our ability to both detect that DNA and also how we interpret the DNA signal that we pick up with the quantitative PCR. So for a little bit of an example of how we're beginning to try and address some of those lingering questions with regards to how we interpret our DNA signal from our quantitative PCR, SWRCB has been involved in a study with the San Diego Regional Water Board where we look at how DNA moves out in the wild with something that we call a spiking study. So for this study, we took a marine mussel and we implanted it into 2 different streams in Southern California, one of which was channelized, and one of which was a quote-unquote natural system. And we wanted to see how the DNA of this foreign organism was going to transfer downstream. And essentially what we found was that when we had this hard bottom, channelized stream, our DNA just flowed really quickly and we could detect it up to 1,000 meters downstream. This is in contrast to what we saw in our natural system, where that DNA really got hung up on a lot of the detritus and organic matter that was suspended in the flowing waters, and so we couldn't detect our signal as far downstream. This is just one example of the type of pilot studies that numerous researchers all throughout both the US and internationally are working on right now to better constrain our interpretation of an eDNA signal out in the wild. All right, so moving on to DNA barcoding. As I said, this method is a really great way to identify the taxons that you have present in your sample. So traditionally you might be used to getting a big jar of your target macroinvertebrates and they're sitting in ethanol and you want to know who some of them are. You pluck one of those species out of your sample and then you search it against a library of known DNA reference sequences in order to identify what species you have present in your sample. So the main limitation of this DNA barcoding approach is that for a number of key taxa, we just don't have those reference sequences available yet in our global DNA library. So if it's not present in our global DNA libraries, we can't identify it in our sample. So this is an area, again, of active research where people are trying to find those gaps in the global DNA libraries and fill them in. Here in California, we have 2 big ongoing efforts, one that's focused on macroinvertebrates and one that's focused on algae, to hopefully fill in some of those gaps in the public repository of these DNA sequences. So first off is a collaboration among a number of labs, including something called the Western Association of Marine Labs, SWRCB, through our the Southern California Bite Sampling Program, in collaboration with the Smithsonian, the Los Angeles Natural History Museum, and the Bureau of Ocean Energy Management. So all of these groups are coming together, collecting a number of different benthic macroinvertebrate samples, identifying them with the help of trained taxonomists, and then sending these samples out for DNA barcode sequencing, and then depositing these sequences, again, in those big global repositories. Similarly, we have an analogous effort for stream algae, and this pie chart here shows you for the number of California algae taxa that we have in what we call our master taxa list, which is approximately 1,700 species. We combed through some of these big public repository and we saw that about more than 50% of the taxa that are on our master taxa list currently lack some of those reference sequences in the global databases. So what we're doing now is working with, again, trained taxonomists to find those species present in our samples, isolate them, grow them up, and then sequence those reference sequences to deposit in those global DNA libraries. We're prioritizing the taxa that we're sequencing by looking at the ones that we know are really abundant across a number of our stream sites, the ones that are key bioindicators according to our algal biological index, and then also the ones that we know really have no sequence relatives within the same genus or family, so there's really a need for generating these reference sequences for these key taxa. Okay, and then the third method I wanted to talk about was DNA metabarcoding. So this is the one that you may have heard referred to as high throughput sequencing, and there's been a lot of activity, especially in the bioassessment field, for applying this DNA metabarcoding technique to environmental samples. And again, the key reason for that is because you can look at thousands of samples, thousands of organisms all at the same time from a single sample. Fortunately, there are a number of universal primer sets, which is how we amplify the DNA of interest, and lots of these target specific groups of organisms, so there are really well-vetted primer sets for bacteria or metazoa, as well as fish. There's some new primers that people really like and have become increasingly popular. But some of the main limitations of this approach, again, we're limited by the availability of reference sequences in public databases. So again, you really need to have the reference sequence present in these DNA libraries in order to identify a species present in your sample. Additionally, with DNA metabarcoding, we get what we like to refer to as relative abundance data. So this is not absolute abundance data. And the relative abundance data that we get, all of the different copies of the individual taxons, genes that are present in our sample can be biased by essentially primer amplification efficiency. If an organism has multiple copies of a gene present in its genome, all of those can either inflate or deflate the relative abundance of a single species present in your results. And so we can't rely directly on the relative abundance measures that we get. We always have to interpret them with a bit of caution. Additionally, across the board, the primers for key groups of organisms, including algae and some myofauna, the best primers to use for identifying those species with DNA metabarcoding are currently unclear. There's no clear winner or favorite DNA barcodes that we should be using for targeting these species using a DNA metabarcoding approach. So again, in California, we have a pilot study that's hoping to answer that question. So over the past 3 years, or 3 sampling seasons, DNA samples for algae have been collected during the bioassessment campaigns all throughout California. And so hundreds of samples have now been collected alongside the traditional morphology samples that always get collected in our weightable streams. Now we have sequenced about 200 of these samples to begin to do some of that primer comparison and compare how different DNA barcode regions perform when it comes to identifying the algal communities that are present in our samples. For a little bit more background on what our sampling and analysis design looked like for this study, we started off with the algae composite sample that gets generated at the end of the stream sampling. And then that sample got split into a morphology sample as well as a DNA sample. The morphology sample was treated as it traditionally is, analyzed with microscopy, a taxonomic list was generated, and then the algal index scores were calculated. In contrast, our DNA sample was extracted and processed, and then we sequenced it with a number of different DNA barcode primers targeting a number of different DNA barcode regions, which you can see listed here. The 16S gene we use for targeting bacteria. In archaea, we have 18S genes that we use for targeting all eukaryotes, and then we have something called an RBCL gene that's very specific to diatoms only. So then we perform the high throughput sequencing using all of these different barcode regions, again generate our taxonomic list, and again calculate our index scores. In this case, we were calculating the new algal stream condition index for our biological index. So the key question we're trying to answer with this study is how do these different DNA barcodes influence the resulting algal community composition, and can we pick a clear winner? So to jump right into the results, some of our key findings from this pilot study. First, the good news. We saw really high repeatability between our DNA replicates. So when we were collecting our samples, we took a number of different field replicates, and we compared how our algal community composition varied, or in this case didn't vary too much, among these replicates in our DNA results. This was a really great finding because oftentimes we don't get a chance to collect field replicates, and if we do, they don't always have such high similarity in the community composition, so this was really encouraging. The next thing that we found was that the choice of DNA barcode that we used for targeting our algal species really did influence our algal community composition. So including up top, an ordination where each of these different points represents one of the samples that we collected, and they're colored according to which of the different DNA barcodes we chose to sequence them with. And then you'll also see that there's some morphology results there. So all the points that are closer together mean the community composition was more similar. If they're further apart, that means that there were bigger differences in the community composition. So we could see that when we were comparing the different DNA barcodes that we chose to use, we saw some really big differences in the algal community structure. Additionally, when we were looking at just the diatom-specific primer set, which as I mentioned is called RBCL, you can see right here in green, because we were using a diatom-specific primer set, we picked up a lot more diatom species, as you might infer, using this diatom-specific primer set in comparison to using what we call a universal eukaryote primer set. So that was, great news. We got really good diatom data, but it begs the question if it's important to be including multiple DNA barcodes when we're doing our sequencing, which increases the cost of doing these analyses. Additionally, this graph here on the bottom shows you the differences in the algal index scores from the different barcode primers that we chose, as well as the morphology data. So the key difference that we saw here for the majority of our samples was if algal index scores were calculated with morphology, they were biased higher, so a higher score than our DNA scores, which were in general biased lower than our morphology-based scores. When we dug into the data, we really wanted to find what was the cause of that difference between our morphology-based and our DNA-based scores. And one of the main reasons was that we currently lack reference sequences in these global DNA libraries for a number of the key algae taxa that we were picking up with our DNA-based approaches. So like we've already heard a couple times now, it's really important to have good coverage of your target species in your DNA libraries. Because we don't, for a number of the taxa we were picking up, it can bias our score. One of the other big reasons that the DNA-based scores were lower was that the taxa that we do pick up with our DNA methods don't always have something that we call our trait attribute data. So these are the ecological characteristics of the different algae species present in our sample that we use to compute our algal indices. If it's a new taxon that maybe we haven't picked up before with a morphology-based method, we don't have this trait attribute information for them yet. And so those scores get biased lower as well. So moving forward, we have a couple of key options for potentially modifying our algal index to be a bit more compatible with this DNA-based approach. We can update our index, which means potentially dropping metrics from the index where we don't have that necessary trait attribute information. We can go about trying to generate that trait attribute information for some of the really abundant taxa that we identified in DNA, again, working closely with taxonomists in order to do so. We can think about grading on a curve where if we can't consistently see that our algal index scores are biased lower by, say, 0.3 in our algal index score, potentially we can just bump up our DNA scores so that they're no longer negatively biased just because it's based on DNA. And one of the other ways that we can go about developing a novel index based on this DNA data alone is with something that's called a taxonomy-free index. And so in this approach, you essentially recalibrate your algal index using your DNA samples alone that have been collected along a stressor gradient, and you begin to assign trait attribute information to your raw DNA sequences instead of a species name. This is a pretty novel technique, but it's one that we're exploring here in California because it really doesn't rely on having to have that trait attribute information to calculate your scores. All right, so I hope that this gave you a bit of an overview and a sense that there's multiple steps in generating useful DNA data for bioassessment applications. So we go all the way from sampling our organisms out in the wild, extracting our DNA, sequencing it, performing bioinformatics, assigning taxonomic ID, and eventually calculating our biological indices. And each of the different methodological choices that we make along this pipeline can influence our resulting biological index scores. So there are a number of different elements along this pipeline that have key decision points that we're actively thinking about to come up with the best approach for generating robust, repeatable data. For example, we might think about what's the best way to preserve our sample or how we extract our DNA. What sorts of negative or positive controls should we definitely be including? How might we be adapting our indices so that they're compatible with this DNA data? So some of these questions are easier than others. Which preservation might we be using? There's been a huge body of literature that's already addressing this. So to have consistent, reliable data, we really kind of just have to pick one best method and make sure that it's getting used across multiple programs so the data is comparable. So in order to encourage greater comparability in molecular-based approaches that are getting applied monitoring programs all throughout the state, we now have something called the California Molecular Methods Work Group, which I think this time last year it was just kind of a glimmer in our eye, and now it is a living, breathing work group within the California Water Quality Monitoring Council. And what's great about this work group is it really was formed to enhance the collaboration and coordination among different researchers across the state for coming up with some of these best practice recommendations and standardized operating procedures for both sampling and analyzing the DNA-based samples to ensure that all the DNA data that's getting generated across the state is of comparable and high quality. All right, so I would just end by saying, the outlook looks good for incorporating molecular methods into bioassessment programs. These methods are already proving to be a nice, rapid, efficient, repeatable alternative to traditional microscopy or morphology-based methods. We're making great progress to standardize them. We have a couple of key areas for further development, which I went over today, including developing additional primers and probes for target taxa, better understanding how DNA moves out in the wild, improving our DNA reference libraries, and hopefully also adapting our existing biological indices to ensure that they perform as well as can be with these DNA-based approaches, and we ensure good data for decades to come. With that, I would just like to say if you're interested in learning more about the Molecular Methods Workgroup, you can head to this link, and if you're interested in joining, go ahead and email me, and I'd be happy to add you to our listserv. And with that, I'll take any questions. Thank you.
Susie, good presentation. What kind of intraspecific variation can you detect with this methodology that might give you even enhanced information on biological diversity that you may not be detecting right now, but that has the potential for giving you even greater amount of information than just the morphology approach?
Yeah, that's a good question, and it varies by species. So a lot of the DNA barcoding work that we do, for certain taxa, you can pick up some of that intraspecific diversity, depending on your gene target. For other taxa, you might have to sequence a slightly different gene or a slightly longer portion of the gene. But the short answer is yes, you can definitely pick up intraspecific diversity, but then it varies by your target organism.
You'll indulge me with one more. Did you do the same kind of study that you did with theology with any benthic invertebrate samples?
Yeah, so in California we have done some barcoding work on invertebrates previously, and there's a number of efforts through like the CABW program, if you're familiar with that, where they're sampling a ton of environmental samples and doing metabarcoding, and benthic macroinvertebrates are in that. For some of our bioassessment efforts so far, because the capacity limitation isn't as extreme for generating the benthic macroinvertebrate data for streams, there hasn't been kind of the same motivation or impetus for testing out some of the metabarcoding. So in California, we haven't been focusing as much on it, but definitely other research groups in California and internationally, especially, the bugs are getting lots of interest for metabarcoding.
That was a really good talk. I was wondering if you've done yet or have done a pilot study in a tidally influenced system?
We haven't, but I think that we are hoping to in the near future as well. So hopefully that's going to be on the docket.